talk about how we create a graded potential, right? So we create a graded potential, which is a reminder that graded potentials are small changes in voltage in response to a stimulus. So there are kind of three ways that we can have these changes. The first is with a chemical or ligand-gated ion channel. What happens, right? So if I've got my plasma membrane, we talked in a previous video about how ions, right, they can't flow through this area. So we need to have a channel that can allow for the ion to flow down or flow out. And so what we have in these channels, right, they can move a specific ion through. There are many different types of these channels, and I want to point out they typically have a particular ion that they want to move in or out to change the voltage of the cell. But how this ligand-gated channel works is, imagine there's a door, and that door requires a key. Your ligand, right, is going to bind to it and function like a key, and so that the door opens. And now your ions can flow either in or out, right? Depends on which type of ion channel you have. Some are going to cause a positive ion to flow in. This is going, if it's positive in, we're going to be depolarizing. So it's becoming more positive inside. If it's causing a positive ion to flow out, it's going to be hyperpolarizing, which is going to make your cell more negative. We also have channels that allow for a negative to come in. That would be hyperpolarizing. And those are also very common. Why we would do this, right? So we want to make sure that we either make the cell more positive or negative to change the response that we want in our action potential. The more positive we get, the closer we get to an action potential firing. I want to point out that we don't usually have a major change with any of these ion flows. You need a lot of change and a lot to be added up for us to actually get an action potential. The second type of channel is mechanically gated. And essentially, it functions like a door without a lock. What you need is some form of pressure applied that pulls the membrane apart. Now I want to point out, when I say pull it apart, I mean like tiny, tiny, tiny pull. Nothing major. But when you think about it in the body, think about, right, if I have a change in pressure in my blood vessels, right, the blood can press up higher on there, and that can change how that neuron communicates to the brain about your blood pressure. Or something as simple as pulling your hair, right? My neurons are not going to suddenly rip apart, but they are going to notice that there was a pull and a tug. And so we can have ions flowing in or flowing out in response to what we want to see. The third type is the voltage gated. Voltage gated channels, right, are often the main one that we talk about when we talk about an action potential, but we're going to see them in other areas of the body. And I want to point out that they still, even for action potentials, we have a lot, we need to have a lot of these voltage gated channels. It's not just one voltage gated channel and suddenly you have an action potential. You're going to need many of them together. So how does a voltage gated channel work? So my voltage gated channel at first, so this is the outside of my cell, this is the inside of my cell. So let's imagine I've got my little door. Imagine a double door situation. So there's two doors to get into my cell. The first door is called the activation gate. Okay, so imagine I'm trying to get from outside inside. And then on the inside, we have what's called the inactivation gate. The activation gate starts closed, right? So we're not letting any ions in, we're not letting any ions out. We are just staying in homeostasis. But when there is a voltage change, right, if there's enough of a voltage change, this is going to cause my activation gate to open. When the activation gate opens, now ions can go in or out. Again, depending on what that transporter is set up like, and it'll depend on the cell. Now eventually, right, that voltage change is going to be enough, right? We're going to have a really big voltage change by just the sheer amount of ions flowing in or out of the cell. When we have a big enough voltage change, it's going to cause my inactivation gate to close, but it doesn't really do anything to the activation gate. This is a common confusion in my class, and the real thing that is important to note is that it's sort of like when you scan your ID somewhere or you're swiping your credit card, you have a specific signal that ties to a specific thing. So my activation gate needs a very specific voltage change, and so does my inactivation gate. They are going to function together but separately. Anyway, it still needs to be added together